This is the second part of the lecture, and now we will talk about the endomembrane system. So, what's the endomembrane system? Basically, this is a set of organelles found in the cytosol which functions for transport of different substances. That would include proteins. So, this membrane transport takes place in the endomembrane system. So, these are organelles that are usually membrane bound. Again, when we say membrane bound, this means that the organelles have their own membranes. So it involves these membrane bound organelles interacting with one another for transport of different substances. Okay? We started first with the endoplasmic reticulum, okay, which is divided into the rough and the smooth endoplasmic reticulum. So this pancake-like structures over the cell can process several cell materials, no? And again, for rough and smooth endoplasmic reticulum, they are different in structure, thus they are different also in function. So we go with the rough endoplasmic reticulum first. Okay? The rough endoplasmic reticulum is found adjacent to the nucleus. When we say adjacent, it's actually near or they are just beside each other. So we have the central nucleus, and just outside of it, you'll find the rough endoplasmic reticulum. And the rough endoplasmic reticulum gets its name from the studded ribosomes around it. Again, these ribosomes are very small and they attach to the rough endoplasmic reticulum, thus providing that rough texture of the surface. Okay? If it doesn't have ribosomes that are studded on it, then that would mean that the endoplasmic reticulum is smooth. Again, the difference between the rough and smooth endoplasmic reticulum in terms of structure is that first, the rough endoplasmic reticulum has or is studded with ribosomes. Okay, while the smooth endoplasmic reticulum is not. Okay? And the second difference is that the rough endoplasmic reticulum is near or adjacent to the nucleus, while the smooth endoplasmic reticulum is found farther. It's actually nearer to your cell membrane, okay, the boundary of the cell. Okay? Now, take note that the rough ER is studded with ribosomes. And again, let's recall what's the function of the ribosomes again? Okay, the ribosomes will function for protein synthesis. So, if ribosomes are attached on your rough endoplasmic reticulum, then that would suggest that it has to involve some protein processes, right? So, the ribosomes will produce these proteins, which will later on enter your rough endoplasmic reticulum. Okay? Why do these proteins need to enter your rough ER? Okay? Take note that the ribosomes will make proteins. They are just there to manufacture them, but they will not do the quality checking. Right? So in order to do that quality checking, they have to be properly processed inside the rough ER. So the rough ER will ensure that your proteins are properly folded and properly processed such that they will achieve their real function later on. Otherwise, they would malfunction and cause some distress inside the cell. Okay? In a society, we can say that your ribosomes are the factories of clothes, while your rough ER could be these personnel who will be folding the clothes properly so they can be sold to your retail stores. Right? While the smooth ER, again, is not studded with ribosomes. Thus, it has no functions that would involve the proteins. Instead, it will synthesize lipids and steroids. Okay? Lipids are a form of fats, okay, including cholesterol. So they will be the ones who will be producing these materials. Now, these are non-protein products. Again, these are functions of your smooth ER. At the same time, your smooth ER is responsible in breaking down alcohol and drugs. Okay? So for those who are, let's say, into drinking alcohol, they tend to have more smooth endoplasmic reticulum inside their own liver. Okay, their cells will have a lot of smooth ER because alcohol is a toxic substance that would need breaking down. Okay, so you have to break it down so you won't feel or say experience the damages caused by this toxic substance. Again, the smooth ER is responsible for non-protein processes. Okay? So, let's go back. No, you have your ribosomes, they produce the protein, and that protein will enter your rough ER. But later on, not everything that would come from the rough ER is actually good. So, they have to be, again, processed for another final touch. No? Let's say, for example, packaging. Right? So, you have folded it in the rough ER, they will be later packaged. And the one that will package them is your Golgi bodies. Okay? So the Golgi bodies are the packaging center of the cell. And they transport and modifies the products that are found or formed inside the endoplasmic reticulum. So your protein is manufactured by ribosomes and then it's folded inside your smooth or rough ER. And then your rough ER will have to deliver it to your Golgi bodies for it to be packaged and be sold later on. No? So for it to be exported, right? So, yan. And how does these products, how do these products travel in your Golgi bodies? So they travel from the cis to trans phases of your Golgi bodies. When we say cis, okay, it's just a term we use that is the one facing your endoplasmic reticulum and the nucleus. It's actually the receiving port, okay? Everything that the ER produces will go to that first pancake layer. And that's the cis layer of your Golgi bodies. Okay? By the way, each layer of your Golgi body is called the cisternae. So that's the cis cisternae. After that, okay, it will be packaged and then move to the next layer and then move to the next layer until it reaches to the end layer, which is the shipping port of your Golgi bodies. We call that your trans cisternae. Again, your cis cisternae is the receiving port. It will receive the products from your endoplasmic reticulum. While the trans phase, okay, this is where the final products are packaged and finally they will be exported from this shipping port. So they will send it out to the cell. They can send it to other organelles inside the cell. Okay? Now, the question is, how do the endoplasmic reticulum and Golgi bodies interact with each other? Okay, how do they send these materials? No, they send these materials through membrane-bound organelles, which we call the vesicles. And the vesicles are very small and they transfer from endoplasmic reticulum to the Golgi bodies, and then they can emerge from the Golgi bodies to the other parts of the cell or even outside the cell, right? So the vesicles are actually like trucks, that will be loaded with these packaged clothing, that were folded already in the ER, packaged inside the Golgi bodies, and their transport will take place inside a vesicle. That's why they are called transport vesicles. So they can send it to other parts of the cell that would need it, or even outside the cell where a response is required. Okay? So I hope that helps you imagine the processes going through. And again, my suggestion is that you can make use of the things found in society to relate everything you know of the things you need to know of inside the cell. Okay? And then we have the cell membrane. It's the cell's gatekeeper, and as mentioned, it's the boundary of life and non-life. It protects the cell from damage, and it's actually the last end where the vesicles will come out, and then the products made could be pushed out of the cell. So it's composed of a semi-permeable phospholipid bilayer, but we will be discussing that in a separate lecture after your first monthly test. So don't worry first about this layer, okay, or about this phospholipid bilayer. And it permits the passage of materials in and out of the cell. So again, when a membrane is semi-permeable, it selects what can only come in and what comes out. Okay? So it can let other sugars enter, but it won't let some materials inside to go out of the cell. So it's very selective in what it should have inside the cell and what should stay outside the cell. Okay? So again, it's the gate of your cell. And we have vacuoles, which are also part of your endomembrane system. Vacuoles are fluid-filled sac for storage of materials needed by the cell. So it can actually store water, food molecules, inorganic ions, and enzymes. Okay? And we have different types of vacuoles. Okay? The first type of vacuole is the central vacuole. And it actually stores that fluid to keep the cell erected. And these are found in plants. Okay? I'll be showing you later a picture or a model of the plant cell showing its very large central vacuole. And we also have food vacuoles that are found in animal cells, such as this diagram here. So they're smaller than the central vacuole. Okay? Again, the central vacuole is only seen in plants. For animals, they have food vacuoles, which are actually temporary structures that would contain the food that your cells obtained from the outside. Okay? So when the cell eats that food, it locks it inside a food vacuole. And the food vacuole will later be digested. So that would mean that this structure is small, at the same time, very temporary. Just for temporary storage of the food. And finally, we have contractile vacuole, which we will also discuss later on in one lecture in this semester. And this actually helps in the osmoregulation and excretion in some protozoans or protists, like the paramecium you've seen already in the lab. Okay? So that contractile vacuole can actually, it's like a kidney inside the cell. And that kidney will function for water storage, and it will burst that water out of the cell so that the whole cell of the protist or the paramecium won't burst at all. Okay? But we will be discussing that later on. Okay? And finally, we have the lysosomes, and these are actually termed as the suicide bags of the cell, but I don't like the term suicidal bags, right? They are actually the digestive system of the cell because they can digest the wastes, that are found in the cell. So for example, if there's a mitochondrion that got tired already and got worn out, the lysosome can actually digest that worn out mitochondrion. So, and actually it can also digest your food vacuoles so that the cell will use the structures that are found in that food to build itself, right? And aside from that, we also have the peroxisomes and glyoxysomes, but we will discuss them more in your class sessions after the first monthly test. Okay? So, in summary, okay, we have here the pathways in which your your proteins are delivered and processed throughout the cell. So take note, for ribosomes to manufacture their proteins, they need to get their message from the DNA. And that DNA will be transcribed first to make your own what? No, that's your mRNA, right? And that mRNA will exit the nuclear envelope, and then they will be received by your endoplasmic reticulum. Okay? The ribosomes first. There should be ribosomes. And then, the ribosomes will make proteins that will enter later on your ER. The ER will fold these proteins properly, such that they will be transported later on in vesicles going to the Golgi bodies. The Golgi bodies will package what the ER has folded. So, say for example, they will be wrapped around in gift packs or in presentable boxes, right? So, Golgi bodies will package them, and then in vesicles again, they will exit your cell membrane, or they can be transported to vacuoles for storage. No? But sometimes, your endomembrane system can also start from the cell membrane when it has to intake something. So, for example, your cell membrane will have to ingest a food material, and they will be locked inside a food vacuole, which will be later digested by your lysosome. Okay? So I hope you get these points on how different parts of the cell interact with each other so that they can process the needed substances for us to survive. Okay? And that ends our second part of the lecture. Thank you very much for listening on the endomembrane system. Our next part will be about your mitochondrion and the chloroplasts.